I'm really excited and happy to invite, and I'm glad that Dr. Clinton Allred accepted our CGIBD speaker today. Dr. Allred is currently the Lake Simpson Dickson Distinguished Professor in the Department of Nutrition at the University of North Carolina in Greensboro. He received his PhD at the University of Illinois Urbana-Champaign, and he completed his postdoctoral fellowship at the University of Kentucky, and then was on faculty, as well as I believe a second postdoc at Texas A&M Intercollegiate Faculty of Genetics and Toxicology. And he moved to North Carolina in 2020. Dr. Allred and I have been actually talking about potentially collaborating for the last couple of months. His work focuses on looking at the gut in the context of diet, the microbiome, as well as host regulation. I thought he'd be a great speaker, given our mutual interests as well as the interests of the CGIBD group in general. And I think it'll open up a lot of avenues for collaboration. So with that, Dr. Allred, the floor is all yours.
All right, thank you very much. Let me see if I can share. You guys tell me if you can see that.
Yes, sir.
Yes. And what I will do is that I will monitor the chat on the side. So if there are questions, just go ahead, feel free to type them up.
Did it swapped screens, didn't it? Or can you still see just the slides?
No, we can see your senior presenter.
Presentation. Okay, hang on a second. Let me try that again. All right, what about now?
Yes.
Looks good.
Okay, excellent. Okay, well, like I said, it's a pleasure to have the opportunity to speak to you. My background is pretty diverse in the sense of kind of my path of getting here. So if we have students in the group, if you ever want to talk, I started out as an animal science student, was working with heifers at the University of Georgia, ended up as a cancer biologist. And when I started my faculty position, I had done most of my work in breast cancer, and including in my postdoc at medical school at Kentucky. And I had done a lot of women's health and estrogen signaling work. And so when I got to Texas A&M 15 years ago, almost 16 years ago now, we wanted to get into a different system. And there was a group there that was quite knowledgeable of diet exposures in the gastrointestinal tract, and specifically in colon cancer. There were experts on omega-3 fatty acids and fiber. And so I came in and we got interested. And so what I'm going to show you today is really kind of what I consider my overview type talk. And so I'm not going to go into a lot of detail in several of these projects, but my goal is, being sort of a new kid to the area and having the opportunity to come into the role I am here at UNC Greensboro, is to sort of see if anybody's interested in what we do, maybe make some connections. So if you want to know anything more detailed about any of these projects, please reach out to me. My information's at the end, and I'll be happy to talk to you more. But this is to try to give you an idea of what we're interested in and why. So let's see. Oh, it's not letting me. Let's see. It's not letting me change slides. Why is it not letting me change slides? Okay, there we go. So I'm just going to give you a brief overview, a little bit of background, and then I'm going to talk to you about some projects. Okay, so for this group, this is probably not a slide. I'm going to have a couple slides in here that for this group it isn't really necessary, but what I'll highlight here is my research group is primarily interested in intestinal epithelia. So a lot of what I'm going to talk to you about today focuses on intestinal epithelia. We have gotten more recently, like a lot of people, very interested in the microbiome. I'll show you a little bit of information about that, but in our more recent projects, we're interested in some other things as well. But my strengths are my my disclaimer here at the beginning is I'm neither a immunologist, gut immunologist, nor am I a microbiologist. So while I will show you a little bit of stuff in those environments, I collaborate with people who are really good at those things as opposed to being good at those things, because we're more interested in signaling within the intestinal epithelial cell. So that'll be the focus of most of this. All right, and so we are interested in estrogens in the colon. And so why is that? That may be something that most of you don't really think about. If you look, and this data is pretty old now, at the overall risk of colon cancer development in men and women, it's about the same over the span of a lifetime. However, if you age match men and women pre-menopausal ages, so in their 40s, women are at a much lower risk of developing colorectal cancer. And that and some other sort of anecdotal things got people interested in what estrogen was doing in the colon. And I'll say that it sort of makes sense. So if you know anything about that physiology, estrogen, after being produced, does go through intrahepatic circulation, and quite a bit is reabsorbed in the lower intestine. And so it would make sense then that exposures to of intestinal epithelial cells, particularly colonic epithelial cells, could be quite high compared to some other tissues. So there's a lot of reasons to be interested in the colon epithelia being estrogen responsive tissues. So I've kind of already talked a little bit about this, but one of the other things that launched this is the Women's Health Initiative study, which focused on hormone replacement therapy. And we don't talk about it quite as much anymore because I mean, they knew this going into the Women's Health Initiative study, which is if you looked at hormone replacement therapy, specifically estrogen therapy, but it was combo of estrogen and progesterone therapy, that it had some real positives. It helped protect against osteoporosis, and I'm going to show you the next one in a second. But it was associated with increasing risk of breast cancer. Interestingly enough, some cardiovascular events came out of that, and that ultimately is why they stopped the study halfway through. It's a huge trial that had populations of women on hormone replacement therapy and a huge population of women not on hormone replacement therapy. And what they were seeing is breast cancer risk had increased, which was not necessarily surprising, but so did these cardiovascular events. And some post-analysis actually showed, interestingly enough, and this relates a little bit to signaling that I'll talk about in the colon, that the primary group of women that ended up having heart attacks or other cardiovascular events were women that had reached menopause and had been menopausal for a number of years and were then put on hormone replacement therapy, as opposed to women who were perimenopausal or just reaching menopause and put on hormone replacement therapy. They saw a divergent effect, suggesting that physiology changes in the absence of the hormone, and then we'll respond to it when it shows back up. And I think we believe that that's potentially the case with estrogen in the colon. So this is small even via Zoom, but this just shows some of the premise behind why we were interested in estrogen in the colon. The overall risk of developing colorectal cancer was lower in the first part of the study, where women were actually on hormone replacement therapy, and then then they stopped the study because of these adverse effects. And I guess since they already had the funding, they followed up a number of years later. And what was interesting is the risk had become identical to the placebo effect. And what this meant to us is that the hormone needed to be around to be protective against colorectal cancer development. So that sent us into this sort of overall objective of understanding what estrogen does in the colon. And this became a major focus of my group, and with the goal of exploiting its benefits of being there and as far as colon cancer prevention. and then without having the negative side effects. That was sort of the pie in the sky goal. And if you're not familiar with estrogen signaling, this is true in most responsive tissues. This is considered the classical pathway of estrogen signaling, where it binds its receptor, it translocates to the nucleus and ultimately causes changes in gene expression. So this is the primary thing that I'll show you today as it relates to ER alpha and ER beta signaling. But there is a third form that is a ER dependent pathway, which is the membrane-bound estrogen receptor, or GPR. And we do study this to some extent with a collaborator as well. This is an area I'd like to probably do some additional work in, but I'm not going to show you any GPR data today. Okay, so the other thing, just as a side note, because I put dietary in the title and then decided this group may not be as interested in sort of the diet work that I do as much as other people, but we do spend time identifying novel phytoestrogens. So this is conceptually why we're interested in it. This is estradiol, and it's opposing hydroxyls. So if you look at the gold standard, which is what all of my graduate work was in breast cancer, genistein is a soy-derived phytoestrogen, and it can bind really all forms of the receptor. It has preference for ER beta, and it's a pretty good ligand. It's not near as strong as estradiol, and there's a whole host of them, and they affect a bunch of, much as estrogen does, they have quite variable effects in the body. So we've done a number of projects where we found novel phytoestrogens and described sort of known compounds as well. So if that's interesting to you, we certainly can do that. But I wasn't going to spend a lot of time. Instead, I'm going to talk a little bit about kind of where we got where we are today. And we started with sporadic colon cancer because all of the data I've shown you in these large clinical studies and even anecdotal data really pointed at sporadic colon cancer. And so we just as a reminder, again, this audience, I don't spend much time on these slides because you guys probably know a lot of this, but one of the great things about studying colon cancer is that we have these distinctive stages up to adenocarcinomas. And in the models, preclinical models, which is what I primarily focus on, we do have these aberrant crypt foci, and there's some argument of how predictive they are that you'll get all the way to an adenocarcinoma. But we believe a lot of them will, and we've done enough studies along with other people to suggest that's the case. So you're going to see me present data that has ACFs, and that's what that'll be referring to. So our some of our oldest data was a cancer research paper that in essence reproduced some of what we were seeing in clinical studies, and it suggested for the first time the knockouts here are estrogen receptor beta global knockouts. So this was quite a while ago, and not much was known about the role of this receptor in the colon. There was only one other paper from Jan-Åke Gustafsson's group, who is credited for finding ER beta. And so what we were able to show is that, sure enough, if you treat animals, if you induce in an AOM model sporadic colon cancer and you treat them with estrogen, you can suppress the formation of these pre-malignant lesions. You lose that effect in a knockout animal, suggesting that the animal needs estrogen receptor beta. I didn't say it, but colon epithelia, as compared to, say, ovary cells or even mammary gland cells, almost exclusively express ER beta. So it's a very ER beta driven system. And so we see that in these high multiplicity ACFs, that ultimately these are the ones that we believe will become tumors. So we see this dynamic effect that we're able to reproduce what we see in people. We then went on to publish a couple of different papers in vitro and in vivo that ultimately shows that this appears to be through a p53 mechanism, and this will be one of those places I don't show you a lot of the data, but the players here are that ER beta needs, in at least in part, p53 to cause this effect. It's an MDM2 related mechanism, and ultimately, we feel like we started to work that mechanism out. So again, I could expand on that. But then we move from sporadic colon cancer into inflammation-associated colon cancer. And again, this particular audience probably knows the reason for that is individuals that have the two major forms of IBD and Crohn's and ulcerative colitis are much more likely to ultimately end up with colon cancer. And so we wanted to understand estrogen, and I'm going to show you some data in just a little bit. Estrogen mimics other environmental exposures that will mimic the effects of estrogen in this system. And so again, this audience knows Crohn's and ulcerative colitis have very different manifestations and pathologies. And interestingly enough, what I've shown you, as we see ulcerative colitis here, and what I've shown you so far is that in sporadic colon cancer, we see a clear estrogen effect. And so when we went to a model here, which is a combination of DSS, which induces a pathology that at least is somewhat associated with ulcerative colitis. It's debatable whether it's a true UC model, but it's used as one. We combine that with carcinogen. You get most people do this model because you get tumors really fast. So you graduate students out there, the first data I showed you, those, in order to get to tumors in the sporadic model with AOM, it's usually about a year. And so you're talking about a whole lot of commitment from graduate students to wait on data for a year. So this model will give you tumors very rapidly. The problem is most people use this model just to get tumors, and they don't appreciate that the tumors that are formed are quite different, and the path to get to those tumors is very different. So we tried to use it in what we believe to be the right way to ask a more clinically relevant question. And in this model, we see that estrogen still suppresses, this is the isoflavone, but estrogen still suppresses the formation of these in both ACFs and high multiplicity ACFs. We went on to look at cell proliferation and apoptosis. And again, this appears to be an estrogen receptor beta knockout mediated effect. And I'm sorry, and then here in this model, we go all the way out to tumors. And so again, we see the same thing in tumors. But the reason I let this in here, because I thought this group might be interested in what we saw beyond that, because we never really expected this. So we saw that the number of tumors, and I should also point out, we always use a pathologist. So we type every tumor that forms, every mass that forms in our animals. A lot of people don't do that either. And so these are true adenocarcinomas. And we see the same effect that we saw before. Now, what was interesting is, however, when we started looking at some of the other markers that we liked, sort of general physiological markers of proliferation and so on, we started to notice that because we were at a much further stage out, we were starting to see a little different physiology. Typically, estrogen doesn't change cell proliferation in a healthy tissue all that much, but we were seeing a bit of induction in an uninvolved part of the tissue and in the tumor. And I can't tell you how this affected our ability to publish this work, because what we were suggesting is there's less tumors, but there's more cell proliferation in them. And that's not an easy thing to sell to reviewers. And so we went back and we did some more work. And this seemed to be the thing that was really happening. So if we look at expression of estrogen receptor beta, and this has been shown now in some clinical samples, but I think there's a lot more interesting questions to be asked about life stage and those sorts of things. But In normal tissue, you get a level of expression of ER beta. It goes down even in uninvolved tissue if you look at different stages of development, so like ACF stage. But then in the tumor, it's getting quite low, and in the estrogen-treated animals as well. So estrogen receptor beta is going down, and estrogen receptor beta tends to be an anti-proliferative signaling pathway in most epithelial cells. But what was really interesting, and what we're one of the only people who have reported it, and the error here is huge, but the reason why is all of a sudden we went from having absolutely no estrogen receptor alpha to like flipping a switch and having these massive amounts in a lot of the tumors. So what we think is happening is as these types of tumors form, maybe less so in sporadic, but as these types of tumors form, they appear to go from a anti-proliferative ER beta type signaling to a proliferative ER alpha signaling. And even though we had less tumors, and in general, they were smaller at the time when we collected them, I think if we had waited, they would have gotten much larger than in the estrogen-treated animals than in the control. So it was an interesting phenotype, and it's started to bear out a little bit in the clinical literature. Okay, so that's where we were, and I'm going to check my time here. So the other thing about estrogen in colonic inflammation is, so that was a tumor story, and we got something we really didn't expect. The other thing we didn't expect is when we were doing that study, and again, a cautionary tale to our graduate students, is when my graduate student, this was right near the holidays, I remember it very clearly, first exposed those estrogen-treated animals to DSS, a large portion of them died quite rapidly. And so there was this literature out there, and there was only one pathology paper at the time published, but there was always this anecdotal thing that in pregnant women with ulcerative colitis, they tended to have more episodes, and in pregnant women with Crohn's, they tended to have less episodes. Now, a lot of things change during pregnancy, a lot more than estrogen, quite obviously. And it was anecdotal, but what we were seeing is in our model of ulcerative colitis, estrogen appeared to be causing real problems. And so I won't go through that for you guys, but we had this conflicting epidemiological data. And this is what I was telling you about, 100% mortality in 48 hours in our study, in my graduate student study, Cameron Armstrong. And we weren't able to publish that either because we couldn't explain it. We couldn't get a pathologist to get to tissues fast enough because it was the holidays. And so we had all sorts of issues. We never expected it was going to happen, but it ended up leading us to doing some interesting work in a whole nother area. So that paper's published if you want to see it. And we've done, since then, we've done some more acute exposures looking and comparing Crohn's versus ulcerative colitis. But what it led us to is asking the question, if that's true, if estrogen for patients with ulcerative colitis, if estrogen might be potentially exacerbating it, and maybe it's in acute periods, maybe it's in chronic periods, what would happen if we're constantly being exposed to something that's estrogenic? And would individuals with ulcerative colitis potentially be a population that is going to be more sensitive to that exposure? And that brought us to BPA, which is a plasticizer. It is something that all of us will be exposed to today. It's all over the place. It is, and it's a known xenoestrogen. So it binds all forms of the receptor. I showed you earlier genistein and its opposing hydroxyls. We have the same type of thing here with BPA. And it's been known for quite a long time. It's been studied in particularly reproductive biology, but less work had been done in intestinal questions or certainly in intestinal epithelial questions. So we set about doing a project in this, and this is a typical design for us. And working with an environmental compound like this, particularly since it's in plastics, is a bit of a pain in the rear because you have to use glass everything and that sort of thing. But what we're looking for and we're trying to do here is just model an acute inflammation event and then look to see if BPA actually worsens that event. And basically, and again, this is published, so I won't spend a lot of time on it, but if you just look at mortality, animals that we were given DSS and BPA, we lost a lot more of them during their recovery time. And that coincided with an enhanced disease activity index. And again, we involve pathologists in doing this evaluation, so we feel very confident in those changes. And it was also associated, if you know, the ways that you evaluate this is weight loss and the recovery from that weight loss. And so pretty much across the board, we saw that BPA was worsening these outcomes. And then interestingly enough, when we looked at inflammation scores, it tended to be regional. So it was really the middle of the colon where we were seeing the vast majority of the changes in inflammation. We expected it to be more significant, first of all, and we expected it to be broader, but it tended to be a bit regional. And, you know, it raised a lot of interesting questions about that. Now, the other thing we did is, in another area that we've gotten very interested in is how the microbiome and metabolites related to the microbiome change. And so this sort of was our first entry into a collaboration related to the other project I'm going to tell you about that has been a very good one. And I work with a group, two actual chemical engineers, one's an expert in microbiology and the other one is an expert in predictive metabolomics, and they have a really nice Nature methods paper out there where they looked at these tryptophan metabolites. And basically the idea here is that the tryptophan metabolites will create a phenotype or a fingerprint of what the microbiome is doing. So they'd be reflective of ultimate changes in the microbiome. So this was our first foray into working with those guys a little bit, and we measured some of these outcomes that because they sort of have a set of these that they believe reflect microbiome phenotypic changes as opposed to just populational changes. And so this is some of the ones that changed. And this one's really interesting. So I won't go into our whole rationale for why we think the ones that are changing, but serotonin's an interesting one because there are a number of people and some really beautiful work done out west looking at serotonin changes after BPA exposure. So our data actually confirm that work and what it does with brain function and that sort of thing. So we do see changes of these, even in the absence of inflammation. We see some different changes when inflammation occurs. So we do think that it's a bigger picture than just simply it's an estrogen and it's causing estrogen signaling within the intestinal epithelia. Okay, so that's that type of work that we do and all the estrogen stuff. The other major arm and project that we're really interested in is the aryl hydrocarbon receptor and colon carcinogenesis, as well as some work that I'm not going to talk as much about, but also associated with sort of normal, if you will, colon physiology. So the AHR is the aryl hydrocarbon receptor. It was originally identified as so it was called the toxicology receptor, because it was known to bind a number of compounds that we get exposed to, toxicants that we get exposed to in the environment. And it also is a nuclear receptor. It had its heterodimeric partners aren't, and it has its primary outcome that you can measure easily is the induction of CYP1A1 and 1B1, or cytochrome P451A1 and 1B1. This is sort of the classical thing you would do in a toxicology, and I didn't say it, but I'm actually also trained as a toxicologist. So and it influences growth and differentiation. Now, the thing about AHR is we're going to call it a promiscuous receptor. It binds all sorts of things. It also has very specific effects in the cell type in which it's being bound. So what might happen in a colonic epithelia might be very different than a mammary gland epithelia, for instance. So AHR is a little bit hard to nail down, but after being identified as this sort of toxicology receptor, much more recently, and one of my collaborators that I've known since I was a pup in graduate school is Dr. Stephen Safe, and he really is one of the grandfathers of AHR. And he started postulating, he and others, that AHR probably wasn't just a bad guy. And there's been a real big movement recently for AHR to be sort of a sensing molecule in the gut environment to bind dietary compounds and microbial derived compounds. And so we sort of entered that fray, again, focusing on what it does in the intestinal epithelial cell. So this is a complex slide, and it shows a lot of these things that AHR does. And what I really want to focus on here is that the ligands, again, it bounds, TCDD is its primarily toxicology ligand. But then there's these microbial things, and this should look familiar from some of the things I was showing you on the other slides. So there was a whole group of us, we called ourselves the AHR group, with Dr. Safe being sort of the top of that pyramid, and we were interested in these microbial derived compounds as well as dietary, which is you may see DIM pop up in some of the data, and if you do, it's a dietary compound for AHR. So why AHR? And I'll go quickly through this, but if you knock out AHR, you do get colon tumor formation, and you can do it in the DSS-AOM model as well, which is the model I showed you for inflammation-associated colon cancer. You get more in the knockout. So there's a clear indicator. Now, in our case, what we were interested in, again, is knocking it out specifically in the intestinal epithelial cells. And there was less work at the time done in that arena. More had been done with global knockouts. And so we were asking pretty unique and individual questions. In this case, because I am in the Department of Nutrition, we did it in the context of a low-fat versus high-fat diet, because again, a lot of the interest in AHR is it being sort of a sensor for compounds that are going through the gut. And we know that being on a high-fat diet it alters what is going through there, probably by altering the microbiome pretty significantly. All right, so in this study, similar, this is more of a sporadic colon cancer model. So we're doing quite a few injections of AOM, and then ultimately those ACF are the outcome. So if we knock out AHR only in the intestinal epithelial cells, so not globally, we do get an induction of ACF in these animals. It's exacerbated in a high-fat diet animal, and as more so than it is in a low-fat, and so that, and again, this work is published as well, seem to fit the idea that AHR is a good guy in this model. We do see some changes in proliferation a little bit. They're more modest, to be honest with you, but this system, when you're not looking within the tumor, you don't see really dramatic shifts in proliferation. So modest ones over time can have a lot of physiological effect. And I'll note here that a lot of the time we see this in the bottom part of the crypt. We will analyze all those. And so one of the, Rob Chapkin, who's a collaborator at A&M, does a lot of stem cell work, and so his group focused a lot on what happens specifically in stem cell populations. I won't go through this, but basically we do get a very pro-carcinogenic fingerprint when we look at gene expression, particularly if we do it within the epithelia. This was mucosal scrapings, but we do get a very pro-carcinogenic gene expression as well. And so this group probably knows, so the last part I'll show you here is a little bit about some of the other analysis we did, looking more at the microbiome. And this was one of the first studies. Now, since then, we've done a lot more interesting things as far as doing fecal transplants that were actually based off of this study. And we've gotten into some more, what I believe to be much more advanced and interesting questions. But this was sort of our first step into this. And we did this study, even given its length and everything else, with the intent that we could get well thought out and planned experiments done where we could trust the the bacterial analysis that we would do from it. In other words, we did a lot of we tried to get all of the animals to a pretty much similar baseline microbiome before the experiment started. We were very careful about going between cages. So all of this data was done in conjunction with a group that really helped train us on how to do this right. So I feel, good about how we did it, though I will say the entire field sort of shifts constantly and what is best practice for collecting data to do microbiome analysis. Okay, so this is, fecal samples. I think this is the termination endpoint for this study. And what we found is we saw quite a significant shift in AHR knockouts. Now, I'll remind you, this is just losing AHR in the intestinal epithelial cell. So we also published a paper years ago that the loss of estrogen receptor beta changed some some bacterial populations. But at the time, in working with the microbiologist that I was working with at the time, he was doing very targeted analysis and looking at specific bugs because people didn't do 16S as much at the time. It was very expensive. So this was more of a global analysis, but conceptually, if you think about it, losing a nuclear receptor in an epithelial cell, and that being the only change, having dramatic effects on the microbiome is still conceptually pretty interesting. So what changes here is you do see an increase in bacteroides and firmicutes. And again, this is not my specialty, but these changes don't always get people really excited. What was really interesting is that in wild type animals, we had this large population of microbia, and it just was gone in our knockouts. And this was absolutely reproducible. So this was a breeding colony, and we could find that in the knockouts, we consistently lost this population. And so why do we care about these? Akkermansia was at least when I originally created this slide, it was the only real member that we knew about being in this group. I just saw some stuff recently that new members are, I think, being identified. But this is sort of the belle of the ball recently, and as of the last 5 to 7 years, a lot of people have studied Akkermansia. It's associated with a lot of things. It is a mucin degrader. It's an indole producer. And so we believe that these were groups that made sense. Now, of course, this is a very global analysis. We're doing much more sophisticated analysis on this now. Again, what I would argue is you see data like this and you see microbial changes. What we want to do that is, I don't know that it's that different, because I think a lot of people understand this and agree with it. the reason we tend to focus on metabolite changes and what we believe are sort of fingerprint phenotypic changes is because a lot of things will change what bugs are there, but ultimately what matters is what those bugs are doing. And so, so these were interesting changes though, and it's some of the first that ever looked at what this environmental exposure was causing. And then the other thing I would say about the work that we do, and this is a bit of a plug, for anyone who's out there that's interested in sex differences, that's a lot of what we do and how we try to collaborate and bring things to the table for our collaborators. I have a long history of looking at hormone signaling in these systems and the nuances that go along with that. There's not a lot of people that study estrogen in the colon, for instance, and what it contributes. And so with NIH wanting more to explore gender differences, people now will write that into grants. So sometimes they compare, sometimes they don't. And a lot of times they just report when they see differences, but they don't necessarily take time to go on and try to explain that. So we're happy to collaborate with individuals that might be interested in asking those questions. We have a way of manipulating systems in order to try to ask specific questions. So I mostly left this in just to highlight the fact that we do see differences based on gender as well. And we've got more data coming out that will highlight that as well. I didn't put it in here because I didn't have a lot of time, and it was good instincts because I want to leave enough time for questions. But one of the things that we're already talking about in collaboration is we also are very interested in what we found is in this last group of animals, these AHR knockouts, is we had a pathologist who was in training. She was getting her PhD, but she was already a board certified pathologist. And she said, you have these lymphoid structures in these tissues, and you should look to see if they're different between your groups, because she was blinded and everything she was analyzing. And so we went back and started looking at this, and there was a paper out there that showed AHR was involved with the formation of what's called tertiary lymphoid tissues. And some of you guys probably know a lot more about that than I do, maybe refer to them in a different way. In the cancer world, they've become pretty interesting because these are things that form, they're not like Peyer's patches. They form later in life when there's insult and that type of thing. And there's a nice review in Science that talks about how when they are associated with tumors, the prognosis is better. So we have a paper we'll be publishing hopefully this month that shows AHR is involved with that. And we used an inducible model in that case and showed that loss of AHR in a sex-specific way actually reduces the formation of these tertiary lymphoid tissues. So one of the things that we're really interested in doing now is getting collaborations together to look at immune cell recruitment to those and ultimately how all of this sort of interplays in the formation of those as well. So that's one of our future directions. I didn't show you that data, and maybe I should have, because maybe that would have been more interesting than some of what I did show you. But we're going there. But conceptually, I would say, and this is from a grant that we put in not long ago, you could sort of change anything up here. We do dietary manipulations. This happens to be those plasticizers. But a lot of what we sort of collectively do with me and my collaborators, we looked at sort of predictive metabolomics. And in doing so, there's multiple ways that these things can influence particularly if it's an estrogen, it can go in and have direct effects. It can change the microbiome. It can change what the microbiome makes, and then it can also get to the liver and cause changes in liver metabolism. So we really focus on really these two in the middle a lot. but ultimately we can swap out most of these things. So I just talked to you about changing the outcome into the formation of these TLTs, but this is sort of the approach that we take globally with the combination of people that we have. So that's sort of how we go about doing things. So I'll wrap up. I've been very fortunate to be funded throughout my career. Early on in American Institute of Cancer Research and American Cancer Society, later through some R01 funding with the National Cancer Institute. I've talked about collaborations, Dr. Arul Jaramaran, who is at Texas A&M, and his main collaborator, Dr. Kyung Lee at Tufts, make up sort of our current team to approach what I was just describing there at the end. Dr. Chapkin and Dr. Safe at Texas A&M. Again, Dr. Safe is an AHR expert, and Chapkin was in my department, and we learned a lot of these colon models from his group. And Dr. Brad Weeks is our pathologist. But most importantly, all the data you saw today, I have some current graduate students, and we're doing a lot of work now. I didn't put any of their stuff in here, which probably would make them sad. But these graduate students have all moved on. Cameron is on faculty at Davis now, and so they're the reason that we have the data that I was showing today, and I have my contact info there. So with that said, I'll put this slide up because I've used it ever since I started giving talks, which is why it looks like it's a scanned page out of a magazine, because it is, but I've used it ever since I started giving presentations back when I was a graduate student. So hence the reason it's about breast cancer. But I'll leave that up, and I'll be happy to answer any questions.
Thank you, Clint. That was great. I'll wait for questions in the chat, and if you have questions, you can either raise your hand or just type them out or just chime in. I can get started. I'm curious. I guess I have a couple of questions. The first is the estrogen alpha data. How are you separating the impact on IECs versus immune cells? I'm curious. I know you've got the, how do you, I'm wondering whether there's a differential or paradoxical regulation in that regard. And I'll ask my second question is after you're done with the first one.
Yeah, so, and the answer is, in that case, we're not. That was a global knockout. But more importantly, we're using immunohistochemistry there, and we think we're looking primarily at epithelial cells in doing so. But, when you look at estrogen signaling in immune cells, including in the gut, I remember one of my graduate students who finished recently, when she entered the lab, and I said, hey, I'm thinking of writing a grant where we start knocking the receptor out of immune cells, and I would still like to write that grant. So just go start summarizing estrogen signaling in different immune cells in the gut. And poor thing, she came back with like a 10-page Excel file of everything she had found, which is to say it's very complex. And I have no way in that data of really making any of those distinctions. I think it's ripe for that. As a matter of fact, I just reviewed an entire paper on estrogen signaling, on influencing checkdowns and immunotherapy in other systems. So I don't think immune cells and estrogen signaling in the gut have been studied near enough to understand. And the problem becomes both receptors are in those cells. So that is not an ER beta driven system. Both receptors are there, and it just gets really messy really fast.
Right. I've also got some questions that they sent me, but I guess you guys can type them on the main chat too. But the mucin in your ACR knockouts, because of the proliferation of the acromantia and I think and the navis, are you planning on studying the mucus layer in the colon to see whether or do you have any indirect evidence that something is happening?
Yeah, so we did measure it. I have it in so what we did is we took from that study where we did the AHR knockouts and the ACFs, what we did is we kept a group of those animals and kept them on that diet and collected fecal material. And we have another paper where we did a fecal transplant into wild type hosts. And in that case, we used antibiotics to suppress. And one of the major outcomes was mucin thickness because we wanted to reproduce that. Now, unfortunately, that paper is going to be tough to publish because what happened is we ended up getting a bloom of Akkermansia in the animals where we didn't want it. And so we got a great Akkermansia effect, and it did change mucin thickness for sure. Unfortunately, it was in the animals that weren't supposed to have it. So it's going to be a bit of a tricky way to make the argument that these were the animals that had the phenotype, you know, and so we'll see what happens there. But absolutely, I think it's an interesting question, and we're trying to answer it. It's just that didn't go exactly the way we planned. We didn't expect that bloom to happen.
And I think one last question with people, you know, we can let them go. I wish you could have met Janelle Arthur. I know she was sort of away with her child, but you know, her AOM model with her paper that was published in Nature, I think looked at microbial dysbiosis in the context of that. And I'm wondering whether what you are seeing in your model, whether there's any sort of overlap with these microbial shifts, and whether inducing injury on top of an ACR sort of knockout, whether that would be something you'd consider to do.
Yeah, I think it's a great question, and I will say also it is, you know, that carcinogen is a secondary carcinogen, so it has to be converted. But we've always worried about exposure for the size of the animal. But what traditionally people who use that model haven't taken into account would be the microbiome, and I think that's a really interesting question, and it probably does explain why you see variation. As a matter of fact, it could explain in part why some mice strains are so much more susceptible to it than others. Those types of differences we've seen for years. So yeah, so I think all those are interesting questions. I haven't done anything in that model for a few years, mostly because we've been so focused on inflammation. But yeah, it's it would be good to ask some of those questions.
Remind me, do you guys have a notobiotic system out there where you're at right now? In Greensboro.
So no, we don't have one on campus. We just don't have enough users.
Right.
Yeah, so
Okay.
we have a great vivarium, but we don't have enough users. So A&M, now A&M does, so I would have access there, but it'd be much better to use facilities here because we do all of, in this collaboration, we do all of the in vivo work.
I think that'd be a great idea. Well,
Yep.
thank you so much for presenting. And when I get emails from people in terms of contacting you for collaboration and work, I'll send them your way. All right.
Okay, sounds great. Thank you all.
Thanks again. Bye-bye. Thanks, Amy.
Thank you.